This video will continue our discussion of Mendelian inheritance. We introduced Mendel's model organisms, the pea plant, in the last video, so let's take a look at how he did his experiments. Mendel's experiments consisted of making crosses between plants with different characters, such as different flower colors, as is shown here. He would do this to create hybrids, that is, pea plants with a mix of genes from two different parents. Left alone, pea plants will self-fertilize, so Mendel would remove the male organs from one flower and then manually transfer pollen from another flower to it. The figure here illustrates this and shows the basic mechanics of Mendel's many experiments. One important precursor to Mendel's experiments was identifying true breeding strains of plants. This term means that when allowed to self-fertilize, true breeding pea plants varieties would produce the same exact traits in these pea plants generation after generation. True breeding plants do this because they are homozygous for the gene that controls the traits of interest. This means that they have two copies of this gene that are identical. This is in contrast to heterozygotes, also called monohybrids, which have two copies of the gene that are different. The figure here shows a basic schematic of how Mendel performed his single factor crosses. He would start with two true breeding strains that produced different traits and cross them. The offspring that were produced were monohybrids, but showed the parental trait that was dominant. In the example here, tall, represented by an uppercase T, is dominant to short, represented by a lowercase t. So already we know some information about these traits because the F1 generation of a cross of two true breeding strains will always yield plants with only the dominant phenotype. If the F1 generation is allowed to self-fertilize, that is, monohybrids are crossed, we see clear evidence of particulate inheritance in the F2 generation, where the offspring are both tall and short. However, these are not produced in equal ratios, but a predictable 3-to-1 ratio, with 75% of displaying the dominant phenotype and 25% displaying the recessive phenotype. So in the 7 characters that Mendel investigated in this way, he saw the same pattern each time. The F1 generation yielded all one phenotype, which was the dominant one, and the F2 generation showed a consistent 3-to-1 ratio of dominant to recessive phenotypes for each character. These results clearly refuted blending hypothesis and supported the particulate theory of inheritance. Before we progress, take a second to appreciate the awesome replication in these studies and just how consistent the ratios he observed in the F2 generation were. So Mendel's work with single factor crosses led him to propose his first rule, known now as the law of segregation. This law states that each organism has two copies of a gene that segregate from each other during transmission from parent to offspring. Each parent only passes on one of the two gene copies. So we look at a schematic of this for our earlier cross of the tall and short plants. We see that in our initial parental cross, each parent passed on one copy of the tall or short gene. In this case, each parent had only one type of the gene, so one parent passed on one tall and one parent passed on one short gene. In the next generation, though, we see that each parent passes on either a tall gene or a short gene, but not both. Determination of which one gets passed on is random, and statistically, there is an equal chance of either being passed on. So, with each parent passing on a tall or a short gene, we can get 3 possible genotypes, 2 of which are homozygous and 1 of which is heterozygous. However, 75% of the potential offspring phenotypes have at least one tall gene, thus, they show the tall phenotype. Recessive traits are only shown when they are homozygous. And in the F2 generation of a monohybrid cross, this only happens about 25% of the time. Understanding this probability is important, and our next topic will focus on that.